your beginning is very fine. Start to boost our students.
Okay, sir.
They can get their future
So,
in technology.
Okay, sir. So I was telling that this is a sketch diagram. This is life cycle of the Homosporous fern. This is the front, and this is the sorai. From the sorai, sporangium is there. When the sporangium dehiscence, so many spores are released, and these spores start germinating. First, there will be filamentous stage, then spatulate stage. I'll talk in the reproductive biology section. Then cordate stage. Then in the gametophyte, there will be formation of the archegonia and antheridia. Then when the antheridia get matures, spores got dehisced, then these spores are released and they fertilize with the archegonia, and this becomes, then young sporophyte is developed, and then it becomes a complete. This is in case of homosporous spore. Likewise, in heterosporous spore, from microsporangia will develop, from and macrospore and megaspore from megaspore, microspore mother cell, from megaspore mother cell, and it will develop the male gametophyte, and megaspore develop female gametophyte, and it will form antheridia and archegonia, and after the fertilization they form a diagram and they form a complete sporophyte. It is in case of heterosporous plant. So why ferns are important? If we consider the economic potential of the ferns and their allies, these include their ornamental value, edible properties, medicinal properties, and some of the ferns are hyperaccumulated of metals and heavy metals and metal oils. Some are biofertilizers, and many are antimicrobial property. So let us talk about ornamental value. As you know, the fronds of the ferns are very interesting because of its beautiful fronds. We can say all ferns have ornamental value. For example, this is Nephrolepis tuberosa. You might have seen in the marriages. This Nephrolepis tuberosa is used in the forming the bouquet, and in the marriages also for in the decoration purpose it is used. Likewise, this is Cyctomium falcatum. This is also beautiful fern. It is it has very good ornamental property. This is Pteris critica. It is also called variegated plant because white strip is there. So this is also another one. And all ferns have medicinal value. This is Adiantum capliborensis. You might have seen this is a very common plant. In the Himalayan region you will find it. And this is Microsome scolopendria. Recently from this Microsome scolopendria, one insecticide protein have been discovered by our proteomics group, which has been published in Nature, which is very effective in the whitefly, against whitefly, from two plants, from Microsome scolopendria and another Tectaria cadunota. I have not shown the picture. And this is another form, Microsorium punctatum. This is also a rare plant, and it has also medicinal property. In all ferns are, you can say all ferns are ornamental value, and all ferns have medicinal property. This needs to be properly evaluated. So this is Pteris argyria. This is also called a variegated fern. It is also ornamental fern. It is growing in our fern house also. And this picture has been taken in our fern house. So this is the succinate leaf. That means when leaves start developing, these crozier leaves, different type of pedal leaves of different ferns. You can see the architecture of this fern. Now, if you might have heard about the tree fern, Scythia spinulosa. These tree fern, big size ferns are there. Other tree ferns like Scythia gigantea, that is also very a rare plant. And this is Doriopteri ludians. It is also very beautiful ornamental plant, and it is highly rare plant. It is not commonly found. And it has also medicinal property. This is Blechnum occidental. Blechnum occidental, when it starts growing, you will find a beautiful pink leaf. When it matures, it becomes green. And this is Asplenium nidus. This is also called bird's nest fern. Sometimes when it gets conducive atmosphere, the frond become more than 1 meter length. For example, in Andaman and southern part of India, you will get these plants in growing nature, and big fronds. Likewise, this is Cheilanthus farinosa. It is called silver ferns. And this is Pterogramma calumnianus. It is also called silver ferns. The back side of the fern, if you see the back side of the ferns, white powders are deposited. It is also another field of research. Why plant is secreting such type of white powders you can say it is nanoparticles powders. If you put in on a paper or in a cloth, the imprint of the plant will come on the paper or on the cloth, the whole front. So this is another area of research. Why plant is secreting this type of powders? What is its requirement? Next is Salaginella plant. You might have heard about the name Sanjivani. Its common name is Sanjivani. It is not that big. If you give the decoction of this Sanjivani plant to a dead body, it can live. It's not like that. Actually, the decoction of this plant, when it is given to a person who, if it is senseless due to heavy hot summer, then if you give the decoction of this plant, then it gets its sense. That's why perhaps its name is Sanjivani. And other group of people have also, they have done research that generally we know that neuron cells do not revive, but it has been proved that when the extract of this Salaginella bacteria is given into the neuron cells, sometimes neuron cells also revive. Perhaps that is why it is called Sanjivani and name is justified. And it is also very it's a drought tolerant plant. You see, in dry condition it is totally closed. All the fronts are closed, and if you can keep this in this condition for 6 months, 7 months like this, and as soon as you put in water, it becomes green. So it's a very interesting plant. So what are the chemical properties? What are the cascading effects associated with that? Is also another. Of course, many works have been done, but still so many things remain to be revealed. What are the process? What are the cascading process that it rejuvenates its life activities? It becomes greens. Now this is Pleopeltis loriformis. This is also a highly resurrected plant. It also when it becomes dry, it becomes coils. But as soon as they get water, they become open, and these are the sites. See the beautiful arrangement of this array. It belongs to the family Polypodiaceae. It's also very resurrected plant. There are so many forms which are resurrection property. In Pyrothea, most of the Polypodiaceae group of forms, they show resurrection property. For example, Pyrothea, other Polypodiaceae group also, and Selaginella family also. Now, if we talk about edible forms, this is Diplogium esculentum. The young leaf of Diplogium esculentum. This is the young leaf of the Diplogium esculentum. It is used as sag in vegetables. In Andaman, it is called pudi sag. In different area, different tribals, their name is different. In Odisha, the Dongria tribes, they call it bhaji. And this is Marcellia. In Odisha, it is called Sunsunia sag. This is also eaten. Now, this plant has gradually. This plant is also getting extinct because of its food value and its highly medicinal value. Angiopteris aebecta. This is called Angiopteris aebecta. In Pachmadi biosphere, earlier so many species were seen, but now because of its medicinal value, now people have exploited from the nature, and very few individuals are found in Pachmadi biosphere region. But in Odisha and Niyamgiri hill regions, in some part in the slope area, I found this plant. But I told to the tribals that you should worship this plant. Like this, otherwise they will destroy the plant. So I told them that it is a favorite plant of the god. So you should not cut this plant. So they are protecting this plant in the name of God. Now, if we talk about medicinal plant, this is Adiantum capillaris veneris, as I was telling. It's a very common plant, but it has tremendous medicinal property. It acts as stimulants. It has a febrifuge means which alleviates, it is an agent which alleviates the fever, which is called febrifuge. And it acts as also expectorant. Expectorant means it is the agent that removes the mucus from the respiratory tract. That means anti-cough property. And it has also purgative. What purgative is nothing but it's a laxative property. That means uh, it just lubricate like thing. So purgatory property is also there, and it is also demulsion. Demulsion means it is an agent that forms the mucilus-like or oil-like substance, which forms a soothing protective over the mucous membrane. So many it acts as the hair tonic, anti-cancerous properties are there. Already so many experiments have been done and it is proven. So these are the chemicals found: tannin, camphorol, persol. These are all the chemicals found in the extract of this Adiantum caplacement. In some tribals, they use as tea. The fresh leaf of this Adiantum caplacement they put in the hot water, and after keeping 5 or 10 minutes, they take the drink. So they use as a tea. Another, this is Adiantum incision. All the species of Adiantum, they have highly medicinal value. The whole plant, it is aromatic, and it is used in different anti-cancer property. It has anti-seisive. Anti-seisive also, this has cough suppression. Anti-tussive means cough suppression. But it helps in cough. So it has also anti-diabetic property. This Adiantum incision. So these are the chemicals found in Adiantum incision. So now it is Adiantum philippense. It is also beautiful ornamental plant. It is kept in the house decoration also. It has a very good medicinal property. The whole plant is used in epileptic. Epileptic means you might have heard about the epilepsy, which is a disorder in the brain, which causes sudden alteration in the behavior. The behavior of the person changes. So in this case, it has good property to cure epilepsy and leprosy, fever, rabies, and ulcers. Likewise, the rhizome part. Rhizome part is the decoction of the rhizome that help in throat affection, and anti-cancer property also there. These are all proven already experimentally. It has proven. So these are the chemicals found. Another Ampelopteris prolifera is a very common plant. Ampelopteris prolifera. This plant has very good vegetative propagation. From the front, one board will be developed. From this board, another plant will be formed. That's why it is called Ampelopteris prolifera, because it proliferates from the boards. So it's a very common plant. So whole fronts eaten as vegetable sometimes, and it has also purgatory property, that means laxative property, antiviral property, and antibacterial property. And these are the chemicals found in Ampelopteris prolifera. Now Helminthostasis jelenica. Now this plant, earlier they have so many plants, so many population are there. Now this plant also getting extinct because of the exploitation of the nature. People are not taking interest how it can be propagated. Simply they are going to the nature and extracting the plant because it has high medicinal value. This whole plant is used as apiarian, an anti-fever, antitoxicant, and sciatic boils, ulcer, and that cures malaria. And rhizome part that help in snake bites, and it has antibacterial property and anti-diabetic property. In this plant, when the rhizome extract with the rhizome were screened out, one chemical called eugonin J and K were found, which has an anti-diabetic property. Eugonin J and K. So this is Lycopodium clavatum. I have taken this picture from the internet. This is Lycopodium clavatum. You might have heard Lycopodium in homeopathy. In the name of this plant, one medicine is there, Lycopodium clavatum. It has also tremendous medicinal value. Whole plant is in headache, aphrodisiacs, that means that is sex stimulant, skin cancer. Katra means it is a term. If there is inflammation in the mucous membrane of the respiratory tract, the extract of this plant help in curing. Like it help in diarrhea, rheumatism. See how much chemicals are there in this plant. So many chemicals are. That's why in Patmani biosphere earlier so many population were there. But last in 2013, when we had visited that area, now people have when the homeopathic people, those who practicing in homeopathy, when they knew about this plant, the population of this plant in Patmani biosphere, they destroy the nature. They exploited these plants from the nature. Now very few plants are there. Very few plants. This is another plant, Lygodium plexusum. It has also very good medicinal property. The whole plant that is expectorant and anti-ovulatory, and it also help. It also work as anti-jaundice. And the fronds has antibacterial property and cough. So rhizome has also anti-rheumatic, anti-fertility, and it is also used in the wounds healing. So these are the in root. What are the chemicals? I did not get the information about the chemicals. It has anti-rheumatic property, anti-fertility property, used in sprains, cabbage, and wounds. Now hyperaccumulator. You might have heard about the hyperaccumulation. The Teres bitata, first Lina Kuma, in 2002, she published a paper that Teres bitata is a hyperaccumulator of arsenic. Then people then came to know that pteridophytes, these are tremendous value, they are good hyperaccumulators. There are so many plants, they hyperaccumulate. For example, Equigetum ramusissimum, all the species of Equigetum, they accumulate mercury. Like I had also done an experiment. I have not shown the image of the plant. One Microsorium scolopendria, that also accumulates chromium in significant amount. Now problem is, we know that these are the hyperaccumulator plants. Suppose there is a contaminated area and we planted that plant, it extracted the heavy metals and metalloid from the area. Now question, how to dispose? If you burn it, again there is contamination of the soil. If you throw it, again there is contamination of the soil. So it is another area of research, how these metals and metalloids we can, after accumulation, we can extract from the plant, and can we convert it to a non-toxic form? This is another challenge. This is Azolla pinnata. It's used as a green manure, which has very good nitrogen fixation property. So now let us talk about some floristic part. So whenever we go, first we should know some terminology. This whole, from here to here, this plant is called frond. In pteridophytes, we call it frond. And from this person to this person, this is called stipe. And from this to this, it is called lamina. And this is called rachis. So these are the basic terminology we should know while going for identification and describing the plant. So this is the Costa Madrid. So these are called pinnae, and these small part, these are called pinnules. And rhizome I have not shown here. On the ground, rhizome are there, and above the rhizome, scales are there. So type of scale, these are the identifying characters. How the frond is, how the lamina is, how the pinnae, whether it is pinnate, then bipinnate. It's a bipinnate plant. In the figure, this is bipinnate plant. So whether it is a pinnate, whether it is bipinnate or bipinnative field or tripinnative field, these are the characters, how the lamina of the blade, we'll know about this. This is type of rhizome. Rhizome, that is also important identifying character while identifying the plants. Some cases it is creeping rhizome. For example, in Polypodiaceae group of plants, you'll find the creeping rhizome. The rhizome is creeping. In some part it is short creeping, some part it is erect, some in caudex. As I have told in, I have shown you that Scythia spinulus are the tree fern. In tree fern, the rhizome is caudex. This type of rhizome is found. So some are medium creeping, some are long creeping. These are the identifying character of the rhizome. Rhizome is also taken while collecting plants. Whenever we go for field trip, we should remember that whenever we collect the plant should be fertile, number one, and we should extract the plant with rhizome, because rhizome is also identifying character. So these are the things which are taken into consider while going for the field trip. Now these are the type of scales. Some are lanceolate, with setiferous. Some are clathrate. These are terminology you have to remember. Some are hyaline. Hyaline means it's transparent. It will see. And sometimes it is peltate. Sometimes it is linear but it dented. Setiferous, bullet. These are the terminology you have to remember. Sometimes it is circular. Sometimes it is linear and setiferous with long spine-like structures. These are the different type of scales. These are the different type of pinnules. How pinnules? Sometimes it is ovate. Sometimes it is caudate. This is called caudate. This is called elliptical. This is lanceolate. This is linear shape. This is reniform. In Regnadium, you will find the reniform type of pinnule, Regnadium. In Doriopteris ludens and Hemionaitis erifolia, you will find this type of structure. This is called sagittate pinnule. Sagittate pinnules. That means just like arrow-like structures. Now, the leaf lamina, the leaf margin also, they are also important of identifying it, whether it is crenulate, that means it is slightly dentate like. It is undulate, wavy-like structure, undulate. Cilia has simple hair-like structure will be found. It is called ciliate. It is denticulate, and some are dentate, big denticulate structure. It is called dentate. Some are called incised, lobed. If your tip is like this, it is called acute. If more acute, then it is called acuminate. Sometimes micron. These are obtuse. If there is not pointed lamina is pinnule is there, lamina of the pinnule, then it is called obtuse. Sometimes it is called truncate. Sometimes it is. These are the characters we have to remember while identifying the plant. Likewise, if the slope of the pinnule in the lower side, then it is called decurrent. If from the upper side of the pinnule, it is called succurrent. So these are the characters which are taken into consider. Now, as I was telling that simple frond, pinnate, bipinnate, this is bifurcate. In Platycerium in bifurcate type of pinnule found, frond is found. If it is single frond, then it is called entire simple frond is called. And if this frond, it is pinnatifid, it is called pinnatifid because complete pinnate is not there. That's why it is called pinnatifid. Here see, it is complete pinnate because complete pinnule has form. Here complete pinnule has not form. That's why it is called pinnatifid. Likewise, here it is bipinnate. Pinnate, then another pinnule developed, that is bipinnate. If this second pinnule is not completely formed, like here, it is pinnate, secondary pinnule is not completely formed, then it is called bipinnative feed. It is bipinnate, but it is bipinnative feed. Likewise, it is tripinnate. Pinnate, bipinnate, then another pinnule developed, it is tripinnate. If the third pinna is not developed completely, like here, it is called tripinnative feed. Just before stage of this, tripinnative feed. So, these are also taken into consideration how the pinna is. Likewise, the veins are also considered. Sometimes in Thelipteris, you find sometimes the pre-veins, and sometimes the fork vein. In Teris, you will find the fork veins. Sometimes first it will fork, then another will be simple fork. This type of venation pattern is also found. Sometimes the lower venlets join together and they become reticulate, means net-like structure. The lower veins, they will unite. So this is another identifier. It is called anastomosis. That means veins form network-like structure. So this is called anastomosis. Sometimes veins will be anastomosis and some included venlets. This is called included venlets. That means one single included venlet. Sometimes the included venlets is branched, as you see here. In Pronephrium, we found included venlets. So this is, you can see here, this is the reticulate venation and these are included venlets and their branch. These are the identifying characters. We should remember. So likewise, these are different type of venation patterns. Sometimes this type of sporangia, that is also important. In some cases, this sporangia will develop in the midrib of the leaf. Sometimes in the veins region, in the linear way. Sometimes in horseshoe shape, as in Diapteris in the horseshoe shape, I will show. And sometimes it forms in cluster. In Osmunda Claytoniana, Osmunda regalis, you will find this type of the separate frond will be there, and this cluster type of Sorai will be formed. So these are some of the type of indusium, how the indusium are there. See in Blacknum, this is the character of the Blacknaceae. In the midrib, you will find the Sorai. In some cases, it is not regular, like in patches. In the midrib, it will found in the midrib, but it will be in patches. As we found in Udwadia. So this is a character of Blacknaceae. That means Sorai will be developed near the midrib. It is a character of the Blacknaceae family. Likewise, in Diapteridaceae, you will find horseshoe-shaped Sorai. Inside the Sorai, sporangia is there. Like in Etheriaceae, you will find this type of diplagate Sorai in curved and linear way. So in Asplenium also you will find the linear Sorai. So these are the arrangement of the Sorai. This is also another important identifying character in while identifying the plant. This shape of the Sorai also taken into consideration. In Adiantense you will find in the margin, it just forms a cup-like structure. Inside that cup you will find the Sorai in Adiantense. Likewise in Teres you will find near the margin. In all Tradaceae group you will find Sorai near the margin. So these are some of the leaf arrangement of the Sorai I have shown. As I told, in Diapteridaceae, it is a plant of Diapteris species. See, this is the Sorai in horseshoe shape. That is also Diapteris. In Scythia, see beautiful leaf arrangement of the Sorai in V shape. Scythia spinulosa, you will find this type of V shape arrangement. This is horseshoe shape structure in Diapteris. I was telling Blacknaceae, you will see. In Blacknum, near the midrib, you will find the Sorai. In Udadia, it is found in patch, but in Blacknum, it is found in the continuous. This is the character of the Blacknaceae. Likewise, in Dicranopteris linearis, here you will find in the rounded shape, and just near the midrib you will find. But it is not Blacknaceae. It is a character of Glyciniaceae. Likewise, in Ethereum, I told that in Ethereum, the Sorai will be found in linear shape, curved and linear shape. In all Ethereum, you will find in curved and linear shape. This is the character of the Etheriaceae family. Now, there are different variations are found because in nature, due to our natural mutation, different type of variation is also found in, for example, in Teres pitata. Different type of cytotypes are there. Diploid, up to pentaploid we find in Teres pitata. So these are some of the locations. It is a photograph in Pachmadi biosphere. In Pachmadi, what happens? You need to get down to the ravines, then you will get the form. But Himalaya, you have to climb the hill. But in Pachmadi, you have to get down to near to the water bodies in the ravines. So this is a photograph myself, and this is a photo, beautiful forms in photograph in Pachmadi biosphere region. So now let us talk about. Now we talked about taxonomic characters. Now let us talk about reproductive biology. First, why we should study reproductive biology? In reproductive biology, what we know, we can get how the plant is germinated from the spore to a sporophyte. We can know how germination pattern is there. If plant is getting thread, why plant is getting thread? If we study the reproductive biology, we'll get some of the evidence why plant is getting thread. So in reproductive biology, the different type of selfing are there. One is intragametophytic selfing, another is intergametophytic selfing, then intergametophytic crossing, intergametophytic mating. What is intragametophytic selfing? For example, there is a Petridis, one gametophyte is there. If from that gametophyte one sporophyte is developed, that means both egg and sperm were developed from that same gametophyte, and fusion fertilization took place, and zygote was formed and new sporophyte was formed. That means in the single gametophyte, all things happen. So this is called intragametophytic selfing. That means in same gametophyte, fertilization took place to produce a sporophyte. Second, intergametophytic selfing. In intergametophyte, suppose in a petri dish, different type of gametophytes are there. Different gametophytes, more than one gamete, not different type of the single species. So many gametophytes are there, and from these gametophytes, maybe from one gametophyte of the Antheridia fertilized from Antheridia sperm will fertilize with ovum with the other gametophyte of the same petri dish. So this is called intergametophytic selfing because it is of the same parent, in the same parent, fertilization taking place in the same parent. Now, intergametophytic crossing, fusion of egg and sperm of gametophyte, each gametophyte originating from the different parietal sporophyte. That means it may be the same species, but gametophyte may be of different, because when in nature, so many populations are there. So the gametophytes may be different from the different populations, but of the same species. So when the fertilization takes place between the sperm and egg of the different gametophyte of the same population but different parents, that is called intergametophytic crossing. Then intergametophytic mating, what is it called? Fusion of sperm, egg from different gametophyte with different gamete origins. That means it may be a different species. Always it is not successful. So generally for successful colonization, generally we believe that intragametophytic selfing and intergametophytic, they play important role for the colonization of a species. Now, for example, I am showing one development part of this is Macrothelipteris torresiana. This is Macrothelipteris. This is the scanning electron microscope of the spore. These are this is different type of architecture of the spore. So this is the whole plant. This is the pinule of the plant. Now, this is we saw this spore in the PNT medium. PNT means Parker and Thompson's. Parker describes some micronutrients and Thompson describes some macronutrients. So in combination, it's called PNT medium, which is very pertinent for the growth of the pteridophytes. So in agar we use PNT solution, PNT medium. So when we saw this spore, first what happened? One protonema will develop and another rhizoid initial. So how the depending on the presence of this rhizoid and protonemal cell, there are different types. For example, here it is Vitaria type because the protonemal initial and rhizoid initial, they remain perpendicular to each other. That's why it is called Vitaria type. So with the passage of time, this single cell become 2 cell, then 4 cell. This is filamentous cell. Then what happens? Periclinal and anticlinal division starts. It becomes a spatulous-like structure. Then so many rhizoids will develop from this from the cells. Now this spatulous type with the passage of time, it becomes cordate shape and a notch is found in between. So with the passage of time, what happens? Below the notch, we find archegonia, and sometimes in the wings we found then antheridia, or sometimes we found near the lower rhizoidal region, or sometimes in the wings. But archegonia generally found near the notch. And depending on the development of the archegonia, if sometimes what happens in some gametophytes, archegonia develop fast, and in some cases antheridia develop fast. If it is the antheridia develop, it will potandrous type. And if archegonia develop fast, then the development of the gametophyte is proganous type. So now, when this antheridia matures, flagellate sperms will be released, but at that time there should be water so that they can swim and they can reach to the archegonia. So after fertilization, they form a sporophyte like this. This sporophyte, with the successful division, they form the complete frond, as you can see here. So this is the electron microscope photograph of one antheridia. When antheridia bursts, this the upper part it dehisces. This is called operculum. It dehisces and spores are released and they fertilize with the archegonia. These are the archegonia structure of the gametophyte. So now, how can you know that this plant is intragametic, the plant is showing intragametic selfing or intergametic selfing. For that, to know that, what happens during our experiment? What we do, we select gametophytes before the formation of antheridia and archegonia, and single gametophyte will be put in a petri dish. For example, in replica, suppose 20 petri dishes you have taken, and each petri dish you have kept a single gametophyte. This is for this is called isolate culture. That means one single gametophyte before the formation of antheridia and archegonia will be cultured. In second part of the experiment, what we'll do, we'll take multiple gametophytes. Here also we have to take care that there is no formation of antheridia and archegonia. So the gametophyte will be taken, and more than 6, 7 or 10 gametophytes will be put in single petri dish. This is called composite culture, for composite culture. Now with the passage of time, each sporophyte develop from the single gametophyte in single petri dish. That means in one single gametophyte we have kept in a petri dish. If sporophyte develop from that single gametophyte, that means here intragametic selfing is there. That means egg and sperm of the same gametophyte fertilize with each other. That's why sporophyte develop. So now we conclude that here intragametophyte selfing is there. Likewise, suppose in some cases, in the single gametophyte we did not find sporophyte, but in composite, in the culture of the same species, in composite culture, that means in a petri dish where we have kept multiple gametophytes. If from these multiple gametophytes sporophyte develop, now we have concluded that it supports intergametophyte selfing. That means it is not supporting intragametophyte selfing. Here intergametophyte selfing is there. That means male part of one gametophyte is fertilizing with female part of the other gametophyte. So this is called intragametophyte. So in composite culture we get intergametophytic sporophyte. We know that intergametophytic is taking place in this. Now, after sometimes what happen, we do tissue culture also. I have not kept the slides. Generally, in pteridophytes, choosing an explant is very important while doing tissue culture. We have tried taking the succinate leaves. We have tried taking the rhizomes. But in fact, even if you go through the literature, you'll find that people have taken the gametophyte as explants. How? First gametophyte will be cultured as usual in PNT medium. Then before the expression of these sex gametophytes, that gametangia, that means antheridia archegonia, before the formation of that gametophyte will put in different media with the manipulation of the hormones. Then from one gametophyte, multiple gametophyte will be, secondary gametophyte will be, then from secondary gametophyte, tertiary gametophytes will be there. Likewise, from single gametophyte, multiple gametophytes are formed. When you will separate these multiple gametophytes and you will kept in different media of the same hormone manipulation, from each gametophyte, sporophyte will be developed. That means from a single gametophyte, at least you could raise 20 sporophytes due to secondary and tertiary gametophytes. So this is the best method for micropropagation in pteridophytes. Of course, hormone manipulation is different in different cases. It varies from species to species. So this is the best way for the micropropagation of hormone, so far I know. Because I have tried taking the rhizome, but I didn't get success. Sometimes what happens, we develop the callus, but when we got the callus and we grow for the organogenesis, we do not develop, it doesn't develop a sporophyte. So in case of form, choosing of gametophyte is very important. That means choosing of explant should be gametophyte before the formation of antheridia archegonia. After the acclimatization, after the acclimatization of the plants through tissue culture, we generally put the plants in the fern house. This is one out of our fern house. You can see so many ferns are growing here. This is another part of the. Here we are growing specific fern. That means Pronephrium nudatum. As I had told you that from Microcerum scolopendria, one new protein have been discovered. And another from the Tectaria coordinata, one new protein have been taken, which is published in Nature by our proteome groups. So for that purpose, we are growing some specific plants for further study. This is another I was telling that that nephrolepis, you can see it in people they use it in hangibas. It is also another fern house in our house. So for ornamental purpose, this is nephrolepis exaltata. It is also hugely used in by the people. Now people of Lucknow are so they are also very fascinated about growing ferns because sometimes ferns are kept as a house plant. Microsphaerum alternifolia and Nephrolepis exaltata. All the species of Nephrolepis they are used as house plant. For 1 week it will be kept in the house. For 1 to 2 days they will kept inside the outside the house so that they will get sunlight. Again after 1 or 2 days you can again keep in the house condition. They don't require that much light inside the house. That in low light condition they can photosynthesis and they can grow. This is briefly about. Thank you so much. Hello.
Thank you so much, sir. Thank you so much. It is strange thing about human. If they find anything useful, they will hunt it till its extinction, and if they do not, then they will cut it off because it is not useful to them.
Hello. Hello. I could not listen. Hello.
Hello, am I audible, sir?
Yeah.
Yeah, I was saying that it's a strange thing about human. If they find anything useful, they will hunt it until its extinction. And if they do not, then they will cut it off because it is not useful to them.
Exactly, sir. If you find in the Himalayan region, if you just take one Adiantum capitis, a very small plant, Adiantum capitis. If you take one Adiantum capitis, it comes associated with a bunch of soil. So their role in soil conservation cannot be denied, particularly in Himalayan, everywhere, not only in Himalayas, everywhere. And these forms, they form the microclimate for the higher plants. So, and many people actually, since they do not form the flowers, so that's why people do not take interest. But their role in conservation of soil is a very important thing.
Exactly. So exactly. So that's why the proper knowledge about this plant, that is mainly mere weed for many people, is very much needed.
Yes, sir,
we need to know its role in every aspect, whether it is economical or ecological.
Yes,
so persons like you, sir, are the flag bearer of such team who actually care about these lower groups of plants. Here another problem arises in this conservation of these lower groups of plants, that unfortunately due to taxonomic misunderstandings and lack of knowledge of the distribution of species outside India, most authors' assessments of endemics and threatened species are considerably misleading in maximum cases.
Exactly, sir. Exactly.
So
in fact, a lot of confusion in the nomenclature also.
Exactly.
Some group of, for example, Cristalla dentata. Some group they are telling Thelipes dentata. Why you are creating confusion? Even in molecular taxonomy, a big group of scientists they have already proved that Cristalla genus is written, but still some Indians they are following the Fraser-Jenkins taxonomy because Fraser-Jenkins has done a lot of work. People Indian taxonomy depend on him. That's why whatever he's saying they are obeying.
Actually, sir, I mean, different classification system, different name. So we need to have the proper knowledge about these plants, and after the proper way of conserving it. And Dr. Bear sir has covered every corner of it in excellent ways. So I am sir personally very much thankful to you because my area of interest is the same.
Okay, sir.
Yes, sir. And this presentation has given a lot of motivation the beginner like me.
Sir, may I know your name?
Sir, my name is Anish Bhattacharya. I am the faculty of Durgapur Government College Botany Department.
Okay, sir. Nice to meet you, sir.
I will definitely contact you, sir, for more suggestions and advices for my research work. So, sir, so the session is open for discussion. I would like to ask the participants if they have any question. As I can see, Professor Mondal is with us and Professor Zahid Hussain is with us.
Namaskar, sir. Mondal, sir. Namaskar, sir. Zahid, sir. Namaskar.
Namaskar, Dr. Vera. It's good to see you after a long time. Yes, it's wonderful to see you after a long time.
Yeah.
And so hopefully you please come to our university so that we can meet. And how is the other colleagues?
Fine.
Fine.
Dr. Manti, Dr.
Good morning. Namaskar to Professor Zahid Hussain Uh,
Ashok, it's been wonderful. Thank you for organizing such a lovely
Yes, actually, one
webinar.
thing I have done a lot of mistake that to invite Professor Jahid Hossain because it was the classical mode and
No,
Behera
no, no,
is
no.
here.
I am here for Dr. Behra only.
You may remain quite busy. That's why I have not informed, and
No, no,
it's
no,
an
no.
apology for me.
Anish invited me, so no. Actually, yesterday I just asked Anish where is the schedule for Dr. Behra.
Yes,
So
yes.
he said it's morning. So
Onis is your student and he is here. And actually, I have not invited you because I think that you may remain busy. That's why. And now, God has helped us to make three of us in a joint platform today
yes,
morning.
yes.
Yes. And after
Yes.
20 years back. After 20 years back, Dr. Behra, Professor Jahid Hossain and myself, three of
Yes.
us have together in a joint platform. And I am
Yes,
so much
we spent very good time at Lucknow.
at Lucknow. And today I see that many things Dr. Behra shared with us, and one wonderful thing is that sort of farmhouse of NBRI that I see in the last slide, and that's why I was delighted to see this thing.
Yes.
Sweet memory, very sweet memory.
Your students can come to our institute. They can visit the.
Yes,
now,
yes.
so many houses are there. For now,
I will make
for dry head conservation. Nowhere you will find in India.
Yes. I will make a trip with our students and colleagues to NBRI, Lucknow. Okay. And one thing that on my behalf, personal behalf, and on behalf of the Department of Botany of Durgapur Government College, we are very much thankful to our Dr. Sandeep Behra. You have presented very beautiful work and the presentation, starting from the normal gametophytic complexity of the fern as well as their life cycle pattern, and ultimately you finish the different aspects of the ornamental values, and particularly you emphasized on the medicinal value of the fern to be explored a lot. And that's why I'm very glad, and we are very much thankful, and we are grateful to you, Dr. Behra, for sharing your extensive knowledge in this particular field. And I am very much thankful to all the participants also, because they have learned a lot of pteridophytic research, and every point you have thrown a particular area of the work, the further research can be done. Each and every aspects you have shown this deliberation in your lecture, and particularly you have shown the future prospect of the medicinal importance, and in the medicine industry how fern could be explored. That also you have highlighted, and that is most important thing. Not only the people normally think that the pteridophyte is the very good and ornamental thing, what can be done on the particular plant materials, but many things could be done, and it has the tremendous prospect in future that you have shown in your presentation. And that's why I think that our students, they will be enlightened, and they will gather an extensive knowledge to have a general idea about the pteridophytes, and they can get their future prospect in this particular field, the pteridology. And again, I am saying to the participants that in India, only in the extensive and the specific pteridology laboratory that is located in NBRI Lucknow, CSIR NBRI Lucknow. So any students after qualifying the NET or GATE or something else, they can take their research career in the particular field, the pteridology laboratory, and they can contact with you in future. Okay, and your lecture is very beautiful, and we are very much grateful to you, Dr. Behra. And another my personal matter, I am very much thankful and grateful to have here with our with my junior friends, Professor Jahid Hossain of the University of Kollani. Although I think that he may remain busy, that's why I have not personally invited him. Although I had a talk that in future we will get together in physical mode to our college, and at that time I will call and I will invite to Professor Dr. Behra also, so that we three can get together in a common platform by physical mode. Okay,
yes.
and I am very much grateful to have our my respected sir, Professor Sudhendu Mondol. I have requested sir to be present over here during the valedictory session, and the respected all judges are also here. Everybody is all of the delegates are invited, and I have the full respect upon them. And my teacher and my PhD supervisor, Professor Sudhendu Mondol, is also here. I have invited sir to share a few minutes only with us, so that our valedictory session may be enlightened with some of the kind presence of my teacher and supervisor, Professor Sudhendu Mondol. Sir actually started our webinar giving his keynote address, a very beautiful keynote address. So now I am requesting to my colleagues and the members of the organizing committee to start the process, carry on. Okay, and I am saying to our students that the students, beloved students, those who are present over here right now, you can ask many things regarding the teridology, current problem and the future prospect. You can discuss with Dr. Behera. He is an eminent teridologist in our country right now. Okay, so that's why thank you all.
Thank you, everybody. Thank you so much. Thank you to the organizing committee for inviting me.
Thank you so much, sir. Thank you so much.
Good morning. Good morning, sir. Good morning,
Yes.
Dr. Behera.
Dr. Behera, I want to ask one question. I am Dr. Ashok Kumar Das,
Sir.
a retired associate professor of a
Yes, sir.
government college.
Dr. Das, Dr.
Uh,
Ashok,
doc-
I'm just intervening you. Sir, you are most welcome in our platform. I could not realize that I over here. Sir, I am Ashok Bhattacharya. I
Yes,
think
yes,
that
yes.
You remember me? And
Yes.
yes, sir.
So I like to
very-
ask one question to Dr. Behera.
Okay, sir.
That is the pteridophytes. You are working on pteridophytes,
Sir.
and lots of informations you have presented here. I have done some works on pteridophytes also. So for the mycology and plant pathology chapter is concerned.
Okay, sir.
One thing I have observed during my research works on pteridophytes, that is in most of the pteridophytes, they have the medicinal value on other diseases, but less report against bacteria and fungi. Do you have any idea why they are not so much effective against bacteria, virus, fungi? They are most effective like stomach disorders, jaundice and any other diseases, but less effective against bacteria, fungi and virus. Do you have any idea regarding this?
Sir, no, I don't have any idea. Here,
Yes. Another things I like to mention here, that if we compare the pteridophytes with the higher plants like angiosperms,
antimicrobial properties there, sir. So here also,
yes, antimicrobial properties, but if
good antimicrobial.
yes, but if you compare it with the angiosperms, it is less. That's why I'm asking you, do you have any idea why it is less effective against bacteria and virus in compared to angiosperms? This was the questions.
No, sir, I
Yes.
don't have any idea.
Otherwise, you have presented a very informative lectures. I have enjoyed your lectures. Thank you so much, Dr.
Thank you,
Behera. Okay.
Hello, am I audible?
Yes, sir. Yes.
Yes, sir. Dr. Behera, it's a wonderful lecture.
Thank you, sir.
It's very informative, and rather I'm thinking I will turn my research career into teridology research.
Most welcome, sir. Now people are vanishing. People are not taking interest in pteridophytes.
Yes, it's very fascinating.
Welcome
It's
to
really
Teridophyte.
fascinating. I have a little question for you.
Sir.
Actually, the Antheridia and Archegonia development, sometimes they're developing simultaneously, or sometimes the Archegonia is developing first and the Antheria is later. So is there any chemical cue between, or the chemical cross-talking between this Antheridia of the Archegonia during the developmental process?
Sometimes one report I had gone through that if Archegonia develop fast, then it secretes one anthrodisian hormone.
Okay.
That that anthrodiogen hormone induce formation of the antheridia.
Okay.
But how far it is to whether it is molecular biology, people they have proved or not. But this type of report I have heard, I have gone through. If ALGAE develop fast, it secretes one anthrodiogen hormone to the media where it is growing. Then that anthrodiogen hormone induces
stimulate
the
that.
formation of the antheridia.
Oh, thank you so much, sir. Thank you. So any other questions from the participant for Dr. Behera?
Students, if you have any queries, you can ask to question. You can ask any questions about the pteridophytes and its future prospect. If you have any questions, you can ask students of our department. Our honorable respected Professor Ashok Kumar Das is also here. He is an eminent mycologist and he served many years in our government service in different government colleges with the very reputation, and he is well-renowned teacher. Many students he has produced. A lot of students.
Thank you, Dr. Vorchas. Thank you very much. Thank you.
Including me,
One
sir.
of his student is here.
I know.
One of his student is here.
I don't know.
Me also.
Me also.
Welcome, sir.
So we are very happy to have in this platform to get our respected and honorable Professor Dr. Ashok Kumar Das. Here he is a very good mycologist and plant pathologist.
Thank you very much. Thank you. Thank you.
International and national level.
Thank you very much. Thank you. Thank you.
Welcome, you are welcome, most welcome. I could not find that's why it is our fault, sir. We are very much grateful to you to get you, sir, in our platform.
তিন দিনই অ্যাটেন্ড করেছেন, আজকে শুধু নয় স্যার, আগের দুদিনও অ্যাটেন্ড করেছেন।
Thank you very much. Thank you. Thank you.
আমি তাহলে তো সত্যি আগে স্যারের সাথে আমি কথা বলতাম, খুব ভালো লেগেছে আমাদের এটা, মানে এটা।
Thank you. Thank you.
স্যার কি আমাকে চিনতে পারছেন? আমি শুভদীপা।
হ্যাঁ
স্যার আমাকেও চিনতে পারেননি, মৌটুসি।
হ্যাঁ, চিনতে পারবো না।
ভালো আছেন স্যার?
হ্যাঁ, ভালো আছি। এখন রিটায়ার্ড তো, এই সমস্ত প্রোগ্রাম কোন কলেজ বা ইউনিভার্সিটি অর্গানাইজ করলে একটু শোনারও আগ্রহ থাকে, জানারও আগ্রহ থাকে।
আমাদের এখানে কিছু হলে আমি আপনাকে পার্সোনালি পাঠিয়ে দেবো।
ঠিক আছে, ওকে।
আমি পার্সোনালি জানাবো। আর টিচারদের স্যার কোনো রিটায়ারমেন্ট নেই, আপনি রিটায়ার্ড নন স্যার, আপনি অ্যাজ ইট ইজ।
একদম।
থ্যাংক ইউ অশোক, থ্যাংক ইউ।
স্টুডেন্ট তোমরা জিজ্ঞাসা করো, আমাদের বেহেরা স্যারকে কিছু জিজ্ঞাসা করো, কিছু থাকলে। স্যার কিন্তু এমিনেন্ট সায়েন্টিস্ট, নেট কোয়ালিফাই করার পরে টেরিডোফাইট নিয়ে রিসার্চ করতে গেলে তোমাকে যেতে হবে ডক্টর বেহেরার কাছে। আর আমরা এক্সকারশন করলে নিয়ে যাব তোমাদেরকে, ওই বেহেরা স্যারের কাছে নিয়ে যাব, দেখবে লখনৌতে বেরিয়ে আসা যাবে এবং ওই ফার্ম হাউস দেখে আসবে, একদম চোখ জুড়িয়ে যাবে দেখলে একদম। এটা এনবিআরআই, আই ওয়াজ দেয়ার and the last 20 years back, along and spent many times with Dr. Behera. Okay, I think they are not asking anything.
I have a question.
Yes.
Okay.
Sir, I am Shubhajit Mullik from Kaliganj Government College, Noida.
Hello.
I am a BSc Botany student.
Okay.
So I have a different kind of question apart from this academic. So sir, when you were a student, what was it which gives you energy, or what was the drive which take you here? Because you are doing research and it need a lot of patience. So what is the drive of your energy?
Actually, honestly speaking, my PhD in physiology and biochemistry. I did my PhD in plant physiology and biochemistry. I worked on a wheat plant. So when I joined NBRI in Teridology group, first 4 to 5 years I was very unhappy that taxonomy, it was not a good subject. It was very boring subject. But when I went to the field trip with my seniors, when I saw the plant in the field condition, then I developed interest. So for taxonomy people, unless you go to the nature, you cannot generate interest in taxonomy. So when you go to the field and you see the plant, what you had read in the textbook, you are looking the plant in the field condition, then that develop interest. So gradually I developed interest in taxonomy and other aspect of the pteridophytes. But during my PhD, I did in plant physiology and biochemistry.
Thank you, sir. I can relate a little bit because recently we had a trip to Darjeeling. Then I got a little bit interest in taxonomy. There we have a sir, Partholud sir, taxonomy specialist. From there,
many plants are growing there.
yes, I get a little bit interest in taxonomy. Thank you, sir.
Okay.
Absolutely, absolutely right, sir. Field is our, or nature is our main mother and the main scientist. Absolutely right. So, any other questions from any participant for Dr. Behra? Then we can move to our next session. And am I audible to everyone? Hello?
Yes, you are audible.
Okay,
Yes,
okay,
yes,
thank you. So here comes the most difficult part of this webinar, at least for me, that is to conduct the prize announcement session. For the past 2 days, we have witnessed beautiful posters, very interesting posters and work of different researchers and scientists from all over India. And today is the last day where we're going to announce the poster presentation prize. And I feel honored to share the achievement and to celebrate all participants' extraordinary work. With excellent performance, all the participants prove that everybody is a winner. So we are fortunate enough to have 5 eminent judges with us who have tirelessly scrutinized each and every presentation, interact with the presenter, and gave them valuable suggestions for their betterment. Please welcome our first judge, Dr. Moshumi Mukhopadhyay, Associate Professor, PG Department of Botany, Bidhannagar College. Her research interest is taxonomy of angiosperm and biosystematics, floristics, biodiversity, anatomy, ethnobotany. Our next judge is Dr. Shubhadeep Sengupta, Assistant Professor from Department of Botany, Bidhannagar College. Her research interest is cell biology, molecular genetics, and plant tissue culture. Please welcome our third judge, Dr. Shobhik Mitra, Assistant Professor from Taki Government College, Department of Botany. He is interested in research like distribution and ecology of epiphytic moss in Darjeeling Hill region, analysis of lipid class, fatty acids, including lipid-derived metabolites for oxalipin pathway and their biological significance. Our fourth judge is Dr. Gunjan Biswas, Assistant Professor from Department of Botany and Forestry with the Sagar University. His research interest is profiling of fungi for different parameters, mainly the pharmacokinetic parameters, evaluation of different nutritional properties. Our fifth and the last judge is Dr. Mohsen Ali Khan, Assistant Professor of Botany, Sidhu Kanu Birsa University. His research interest is Paleozoic and Cenozoic paleobiology using plant micro and mega remains, evolution of plant taxa, paleoclimate, etc. Thank you all for sparing your valuable times for the past 2 days for judging the poster session. As we can see that all our judge panel, all the judges in the panel are quite diverse from each and every nook of the plant science. So now I would like to request our judge to share few of their thoughts regarding the webinar, and because it will enrich us in a best possible manner. So I want to hear few words. Actually, we want to hear few words from our judges regarding this webinar. I can see Dr. Shubhadeep Sengupta is here with us. Ma'am, can you please share some of your words with us?
Yes, am I audible?
Yes, ma'am, you are audible.
Okay, so thank you for giving such a nice introduction. After all, first of all, I would like to thank Dr. Devnath Palit, Honorable Principal, Durgapur Government College, and all the organizers for giving me the opportunity to be the judge of this poster session of this seminar. What I want to say that I thoroughly enjoyed the session. All the participants have presented very well, and their posters were very well organized. All the participants have said in a very diverse way. Topics are very diverse, and I thoroughly enjoyed their topics. And poster compositions were very good. So it was a nice experience for me to judge this poster session, and it was difficult for me to judge all the posters. However, there should be some differences between the posters. So overall, it was very good experience for me, and thank you all for providing me the opportunity to join this webinar. Thank you.
Thank you so much, ma'am. So the winner are pulled from, I guess, no other judges present here with us. Let me confirm once again. No, they're not with us. So then I will move to the next part directly. The winner are pulled from the various branches of plant science. Now we have reached to our final zone, and I want to take the permission of judges present over here for announcing the winner. So, ma'am, can I announce the winner for our competition?
Yes, please go ahead.
Thank you, ma'am. So, without further delay, the third prize goes to Miss Akanksha Srivastava from Department of Botany, Institute of Science, Benares Hindu University, Varanasi, for her work on cadmium stress and recovery in Anabina. And also to Tanushree Agarwal, Miss Tanushree Agarwal from Department of Botany, University of Calcutta, for her work on dihydrogen gene isolated from the moss and use it in agriculture. Akanksha and Tanushree, are you both present over here? Akanksha, Tanushree?
Yes, sir. Yes, sir. I'm here. I'm Akanksha.
Congratulations to both of you.
Thank you so much. Thank you so much, sir. I'm really very grateful to you all.
Okay. Tanushree, can you please share a few words with us?
Thank you so much for selecting me. I was when Shondipanga told me about this conference, I was all eager to participate and share my work with everyone. And it was a great seminar of 2 days. I mean, it was very enriching for me. Attending these e-seminars at least gives us a different kind of experience, like going to the lab, and it becomes very monotonous. So these kind of seminars very enriching for researchers like us, and it motivates us a lot. Thank you a lot.
Thank you, Tanushree. Ms. Akanksha, can you share a few words with us regarding the seminar? Ms. Akanksha, can you hear me? Okay. So I will move to our second position. So the second prize solely goes to Ms. Sudeshna Nandi for her work in a natural derivative from ethnomedicinal mushroom from Department of Botany, Calcutta University. Ms. Nandi, are you present over here? Congratulations, Ms. Nandi. So I guess Sudeshna is not here with us. Okay. Now, the first prize goes to, there is two winner for the first position, and the first one is Ms. Shomanjana Katua from Krishnanagar Government College for her work in 3 newly discovered edible mushroom to tackle lifestyle disease, and also to Ms. Shampa Kundu from Sidhu Kanu Besha University for her work in first Cenozoic macrofossil evidence of polypodiaceae from India. Ms. Kundu and Ms. Katua, are you present over here? Ms. Shampa Kundu and Ms. Shomanjana Katua. So they're not with us, but anyway, congratulations to all the 5 winners. But hold on, there is still more. Now, all the entries are so excellent that we could not resist ourselves, and it will be an injustice if we don't acknowledge 2 such posters that involve excellent fieldwork, survey work, and to encourage the future student in carrying out basic research in plant taxonomy, phytodiversity, and medicinal plant. We have selected 2 posters for special award. One by Mr. P. Santhanapandi from Department of Plant Biology, Presidency College, Autonomous University of Madras, for his work on therapeutic diversity and species richness of ethnomedicinal plant in Somagiri Hills, and to Ms. Presina Rai from Department of Botany, Calcutta University, for her work on variation in vascular epiphyte assemblage along altitudinal zone in temperate forest. Ms. Presina Rai, are you present over here? Yes, Presina is here. Congratulations, Presina.
Thank you so much.
Share a few words with us?
First of all, I would like to thank the entire organizing committee of this Student National webinar and giving me an auspicious opportunity to present a portion of my research work. And I am very much thankful. And that's
Okay, thank you. Thank you so much, Ms. Rai. And P. Santhanapandi, Mr. I guess is not with us, but anyway, congratulations, a heartily congratulations to all the winners, and thank you all for participating in the competition. To all the registered participants for this webinar, sorry, we have shared a feedback link in the chat box. Please submit it as soon as possible, upon submission of which you will receive your participation certificate. We will send the award letter and the poster presentation certificate to all the presenter and the winner in due course through mail. If you face any problem regarding the certificate, please come back to us without hesitation. We'll definitely help you out. Now, finally, we have reached to our last phase of this webinar, and I would like to request our respected convener, Dr. Prashanto Saha, to deliver the vote of thanks for the webinar. Dr. Saha.
Yes, Dr. Roy, I'm just interested. One thing, Dr. Sandipan.
Yes, sir. Yes, sir. Yes, sir. Tell me.
One thing, our most senior most and respected sir, Professor Mondol is here. So sir started our webinar with his kind keynote address. So I'm requesting you to announce and to call our respected sir to say something general comments about our webinar, so that before the vote of thanks, the finishing part could be more enriched. So some general comments only on the basis of our webinar conducted.
Yes, sir.
So you please call to our sir, Professor Mondol.
Pleasure if you can share some word with us.
Hello.
Yes, sir. Yes, sir. We can.
Good morning to you all. I'm very happy that you, the young professors of the Durgapur Government College, have taken the initiative to highlight the academic scenario of the botany or the plant sciences in today's scenario, that is organizing the national webinar, and it will help the young students to judge their future pathway. Definitely, it could have been better if we could have interact in an open session, but anyway, overall it's an impact, and I'm very happy. And you have selected some of the speakers. They are very good. They have delivered very good lecture in different aspects. Yesterday I have also seen the Rosario, Asharata Di Rosario's lecture, today morning with the reproductive biology, from NBRI, and also that yesterday, day before yesterday's lectures. It was really interesting, and it is a thought-provoking. All of them tried their best to focus to the limelight, to the botanical sciences or the plant sciences in general. And so many posters, you have given the award very categorically, selecting. It's difficult for the selectors to judge the merit one or two, or even in the sometimes duplicate also there. It's very difficult, but still you have selected someone who will gain, have the first position or the second position. It is there always in the educational scenario. So it's very interesting, and I would like to congratulate you, and I appreciate your activities, academic activities in the present day context. Thank you very much. I'm very happy, and go on keeping it up for your future upliftment. Thank you.
Thank you, sir.
Thank you so much, sir. It's our pleasure that you were with us at this valedictory session. It's our pleasure, sir. So I can say that Ms. Shomanjana Khatwa joined us. So congratulations, Ms. Khatwa. You are the winner for the first position in the poster competition. So can you share a few words with us, Ms. Khatwa?
Congratulations, Dr. Khatwa from Kishnagar Government College.
Thank you, sir. Thank you, everyone. I'm very honored and very thankful to everyone that you liked our work.
Good worker, I know. You from
Yes. Thank you, sir. Thank you. I'm very, I feel very motivated that you liked our work. That is the main achievement for me. That I have contributed something to the science.
Thank you, Ms. Khatwa. So we will always try our best to contribute something to the nature.
Yes, yes.
What we are taking from the nature, will give back to the nature.
Yes, yes.
We'll try to contribute. So, I want to take the permission from the head of the department. So can we move to the valedictory session, sir?
Yes, yes. If any participants can say anything else,
Yeah,
then
if any participant can share their thoughts, they can, please, their feedback.
So if any participants can't say anything else, then you can proceed. My teacher and supervisor, Professor Mondol, is here, and we are very pleased to have our sir in the last part also, and we are so grateful you, sir, for your presence, kind presence with us.
Thank you. Yeah.
Grateful. Yes, sir.
Thank you.
So now I will request our respected convener, Dr. Prashant Saha, to deliver the vote of thanks for the webinar. Dr. Saha.
Thank you so much. Thank you, sir. And good morning, everybody. It's my privilege to propose a vote of thanks and acknowledge the contribution of all who made this event possible. I, Dr. Prashant Saha, on behalf of the organizing committee of the 2 days national webinar on Botany Through Modern Perspectives, ALGAE to Angiosperm, extend my hearty vote of thanks to the dignitaries who spared time for their busy schedule to grace this occasion. We had an opportunity to hear your thoughts, and this will surely encourage us in our future endeavors. Certainly, the past 2 days were useful, and we all got the opportunity to be aware with the recent developments in the vast field of botany. But organizing such a conference was not possible without appropriate preparation, wise working hands, support from every corner, and blessings. So now it's time to convey our gratitude to all who have extended their support to make this event successful. At the outset, on behalf of the organizing committee, I would like to express my profound gratitude to our chief patron, Dr. Devnath Palit, principal of Durgapur Government College, for his continued motivation. Sir, you are our source of energy. Thank you, sir, for always inspiring us. A special mention to our respected IQAC coordinator, Dr. Abhijit Mondal, for his motivation and for being the catalyst that stimulated us to do our best. I would like to record our deep sense of obligation to the Secretary of the Teachers' Council, Dr. Shopon Kumar Ghosh, for his motivating and dynamic leadership. I humbly extend my thanks to all the faculties of Durgapur Government College for their support. I highly thankful to the eminent members of our National Advisory Committee for their wise mentorship and encouragement. I want to thank our eminent keynote speaker, Professor Sudhendu Mondol, for his active participation in our event. Sir, you really enlightened us. Your deep and intellectual way of imparting knowledge has added to the glory of this event. I thank our plenary speakers, Professor Shobhan Mukherjee, retired Professor, Department of Botany, University of Kullani, Dr. Ashalata D. Rosario, Associate Professor, Department of Botany, Narsingho Dotto College, Professor Krishnendu Acharya, Professor, Department of Botany, University of Calcutta, Dr. Sandeep Behera, Senior Scientist, CSR, National Botanical Research Institute, Lucknow, Dr. Yogesh Mishra, Assistant Professor at the Department of Botany, Banaras Hindu University. Your participation have surely given us depth and deliberation. I'm really indebted to the eminent judges of our poster session. Dr. Mousumi Mukhopadhyay, Associate Professor of the Department of Botany, Bidhannagar College, Dr. Shubhodipa Sengupta, Assistant Professor, Department of Botany, Bidhannagar College, Dr. Shouvik Mitra, Assistant Professor, Department of Botany, Taki Government College, Dr. Mohsin Ali Khan, Assistant Professor at the Department of Botany, Shidho Kano Birsa University, and Dr. Gunjan Biswas, Assistant Professor, Department of Botany and Forestry, Vidyasagar University. Thank you for participating to evaluate the work of our poster participants. We continuously contact our judges with our calls, messages, WhatsApp, and emails, to which they promptly and patiently replied. Thank you, experts, for putting up with us. My overwhelming gratitude to our experts for their suggestions to our participants for improving their research work. That really means a lot. And such scientific interaction is what that underlies the key reason for organizing such a conference. I thank all the participants of our poster session. I've been very impressed and touched by the abilities of the participants of poster competition for answering the questions that were set out for you. And I think that it itself is a difficult task, and all the participants did it very well indeed. After my heartfelt thanks, now we look forward to publishing your abstracts. I must say that all the participants of our webinar were exceptional, because without a disciplined and engaged audience, a webinar can't be successful. Equally, on the personal behalf, I am thankful to my department colleagues, my esteemed joint convener and the head of the Department of Botany, Dr. Ashok Bhattacharya, the organizing secretaries, Mrs. Moutushi Shen and Dr. Sandipon Ray, and the coordinators of the event, Mr. Dibbendu Medha and Mr. Anish Bhattacharya, for shaping this conference so nicely and timely. Especially, I am short of words for my esteemed colleagues, Dr. Sandipon Ray and Mr. Anish Bhattacharya, for their involvement and willingness to take on the completion of tasks beyond their comfort zones even. I would like to thank Mr. Puthik Chandra Saha, the non-teaching staff at the Department of Botany, for his untiring efforts for the betterment of our department. Though last, but most importantly, my special thanks goes to all the students of our department. It's because of you that we are here. I apologize if I unwillingly forget to mention any name. So thank you everybody again, and wish you a very good day. Thank you.
Thank you so much, sir. So this concludes this webinar, and I must express thanks to the team who has helped with staging this webinar and all our participants and guests. We appreciate you being here. Thank you so much.
খুব ভালো হলো, স্যারও আমাদের সাথে লাস্টে একদম থাকলেন তো। এটা
আমি তোমাদের প্রশান্ত সাহাকে আরো ধন্যবাদ দিচ্ছি, খুব সুন্দর করে ভোট অফ থ্যাঙ্কসটা খুব ভালোভাবে বলেছে, কাভারিং অল অ্যাসপেক্টস। একটা ন্যাশনাল ওয়েবিনার ডিপেন্ড করে হাউ ইউ ওয়েলকাম দ্য গেস্ট, ওয়েলকাম অ্যাড্রেস এন্ড দ্য ভ্যালিডিক্টরি, দ্য ভোট অফ থ্যাঙ্কস। দুটি
স্যার আমাদের
হয়েছে প্রশান্ত, আমি দেখলাম, আমি পুরোটা শুনলাম প্রশান্ত, প্রথম থেকে শেষ অবধি তুমি যে সুন্দরভাবে সবাইকে নিয়ে বলেছো, দ্যাটস ইওর এবিলিটি অ্যান্ড ইওর ট্যালেন্ট। কনগ্রাচুলেশন, কিপ ইট আপ, খুব ভালো বলেছো।
থ্যাংক ইউ সো মাচ, থ্যাংক ইউ সো মাচ।
অনেকে জানো তো ভ্যালিডেট এই ভোট অফ থ্যাঙ্কস বলতে গিয়ে বলে আই গিভ মাই ভোট অফ থ্যাঙ্কস, আই গিভ। আমি এগুলো খুব মেটিকুলাসলি হাউ দে স্পিক, ইংরেজিটা বলে এটা শুনি, সেই জন্য আমি খুব ভালো করে প্রথম থেকে লাস্ট অবধি তোমার আই ডিডন্ট ফাইন্ড এনি মিস্টেক। এত each and everyone and explained nicely and proposed your vote of thanks. Fantastic, congratulations. খুব ভালো অ্যাঙ্কার করে। আগের দিনে আগের সেমিনারও দেখেছি ইন্টারন্যাশনাল বায়োডাইভার্সিটি ডেতে